now that we are started, yeah, I want to say that we're going to be talking about the Antibot and what it works. Now, the Antibot is part of this very big, thick paper, and I want to go through the thick paper and talk about how that thing works. There's also a supplemental paper that is this. This is actually how the Antibot works, because the Antibot plays a role here, but it doesn't play a major role, and I want you to have the biochemistry of how it works. So that's what the other handout is. We're going to go through both of those. So this is just a reminder to watch this on the playlist on YouTube when I get it prepared, or you can actually find it from that code. And cancer immunotherapy, 9 years ago, was Breakthrough of the Year. And it's actually doing a lot of things. There are a lot of immunotherapies that do. And if you look up here, what molecule do you see but our friend the antibody binding to a cell and doing something? That appears to be binding to its constant region, so that appears to be some kind of Fc receptor. But it made a big leap forward about 10 years ago, and they acknowledged that. But the problem is, it's expensive. Antibodies are complex molecules. You produce like thousands of antibodies off of like dozens of templates, you know. So it's hard to purify each one. To prove it as a drug, you have to have it as a single chemical entity to use it as a drug. We usually don't like using serum or sera as drugs and things like that. So it's expensive to get these individual things and to make them work, and also each person's antibodies are slightly different, right? And then you make one that works against different things. And so you have the issue of the price of drugs in general has gone up. So immunotherapy is very expensive by its nature. Maybe we can figure out how to do that a little bit better, but there's a lot of it that's going to be sort of part of the healthcare economics is a problem that I'm not sure any of us knows how to solve right now. But the thing about it is that to find these cures and to find different antibodies, you can do a lot of grad student work, but a lot of that work can be repetitive. So to take care of the repetitive work, they set up automatic facilities. And actually in industry, there's a ton of these. And as you know, James Wells sort of crosses the line between academia and industry. So I believe this is mostly academia, although I'm sure they have some industrial partnerships. But this is an academic lab that looks like an industrial lab. It looks like a biotech lab, and they have a robot in the middle. Often you will have like one of these hoods. When we take a tour of a local biotech, you will see things like one of these over in the place, and there will be scientists that will run them from instrument to instrument. What James A. Wells did is he put a robot in the middle of all these, and he made the entire process be automated from beginning to end. And what it does is it makes antibodies with phage display. And that should sound familiar because it's building off of Delano and Wells that we had. By the way, I've been talking about Delano and Wells since I've been teaching this class. But the Antibot is much newer. I never thought that I would be able to tie together my interest in structural immunology with James Wells's experiments. But there is a common theme. There's a common question that I'm asking about how do protein interfaces work. We have a chemical interface that defends you against invaders. How does that work? We can study it by making a bunch of those interfaces and then seeing which ones are the best. And that's, so it is actually connected to this old paper that I always liked, even when it came out. It just, you can't really predict where the stuff that you like in 2000 is going to end up in 2023. So the antibody is actually part of this recombinant antibody network. So you see they're making the proteins recombinantly like we do with E. coli. And we're able to make, we made fragments of a mycoplasma protein this year. You can also make fragments. We've also made immune receptors, human immune receptors. They just don't get the glycosylation they need, but this pretty much works. And so you have the robot, and this is at the University of California at San Francisco, UCSF. There are other things at U Chicago and U Toronto as well. So I want to read through the Wells paper. So I'd like you to get out those papers. I'm going to ask you questions. Now, because part of our class is not here, I'm not going to necessarily call on you. I guess I'll be nice and answer the question myself. If you can answer the question, please do go ahead and do it. And if I ask you a question, by the way, the rule is, if I'm asking you a question and you're just blanking or having a bad day, you can always say pass. If I ask you a question, passing is always an option. Okay. So with that, let's start with the abstract and the title. Okay, the abstract does not mention the antibot, but we wonder which sentence in the abstract was done by the antibot. So yes, I want you to look through I want you to skim through the thing, and then point to the sentence when you find it that says what the antibot did. It's in the first half. Is there a word that stands out to you that you think indicates what the antibot is involved with? What do you see? So they're looking at and where is that? Oh yeah, quantitative surface proteomics. Surface shows you that you can use antibodies to look at the surface of the cell. And they're going to be quantitative, and proteomics means you're looking at everything that is proteomic. So this is what the antibodies are used for. The antibot is how the antibodies are made that are used for this purpose. So this is kind of the big purpose. We want to do a big scale scan of the cell surface in a sense. We're using proteins to do the scan. And the proteins that we're doing, remember the word, actually, if you look at the other handout, what is the big first word that you see is recombinant, right? When you see that word in this, you can say, oh, okay, we generated a toolkit of recombinant antibodies. And they ended up generating them to 7 of the RAS-induced proteins. Okay, so RAS-induced means RAS-driven human cancer cells. So you can actually get from this that RAS, they're following a cancer model in which the protein RAS is mutated, and that's what's causing cancer here. It's a very common cancer mutation, in fact. And do you remember RAS from Biochem? Actually, we covered it at the very end of Biochem 1, so it might have been a while ago, but do you remember RAS signaling, or maybe from another class? Did you talk about RAS in another class, maybe? Do you think I think we talked about it. Cell biology? Yeah, well, it depends on if you're talking about cell growth pathways. It's the central protein in cell growth pathways. If it gets mutated, you're kind of in trouble because the primary switch for turning cell growth on and off is messed up. It's either stuck on, well, stuck on is a problem. Stuck off is not as much of the problem. And look at the beginning, first and last sentence will always tell you the purpose, the overall purpose of the study. This work presents a technological platform for attacking RAS from outside the cell. And remember, RAS equals cancer. So we're attacking a RAS, but RAS is a signaling molecule. Remember, all of those signaling molecules are inside the cell. So you're somehow using an antibody, which has to be outside the cell, to attack RAS, which is inside the cell. And that's where what Savannah pointed out about the quantitative proteomics comes in. They're trying to find something on the surface of the cell that will allow them to recognize a cell in which RAS is mutated. RAS is never going to be on the surface, but it affects the whole cell, so there might be another protein that it affects that will be on the surface. And by the way, when you're looking at the abstract, you do always want to look for the spoilers. You want to see what the end result is going to be. And they end up with antibodies targeting CDCP1. And to be leveraged to deliver payloads to RAS-transformed cancer cells. So you want to look for CDCP1 whenever you see it, because their end result is they found antibodies that will target it, and they found that it is associated with RAS mutation. So the two kinds of cells they use, they're using a very common and very central model for cancer, in which you have two cell lines that are grown up, and you have the mutated RAS, G12V, and it transforms the cell into a tumor. This one little mutation, G12 to V, in that one particular place, makes the RAS be turned on and makes the cell a tumor. You can even tell by looking at these two cells. Even I, who haven't taken cell biology, can look at these and can say to myself, oh, there's something different. And apparently this is what a tumor looks like. And so before they did this, they actually did comparative proteomics to look and see, are there proteins on this cell surface that are always on this cell surface, the cancer cell, and never on the non-cancer cell. That way we could look at the surface and say, that's a cancer cell. And so we are coming into it in the middle of things. And so how do they want to use an antibody? If you just throw an antibody against something, it won't necessarily cause any problem. It'll stick to it, right? But there are some cases in which even just having an antibody will actually serve a therapeutic use. This is the famous one, Herceptin, which Herceptin is one of the first immunotherapy antibodies. And so if you have a breast cancer cell, Herceptin, a breast cancer cell normally has a certain number of HER2 receptors. But if you have too many HER2 receptors, and they send a signal for growth, so it makes sense. A cancer cell might have more HER2 receptors. That might be part of the problem. Herceptin will actually bind those, and because they need to come together and move around to signal, Herceptin locks them into place so they cannot signal, and then the growth signal is not caused. This is actually kind of a lucky break. Not many receptors actually work like this, but all you need is an antibody that binds, and this will work. I want to make sure that we're recording nice and well. When I have this going on, I want to make sure that Jake can hear this. Okay. So remember, Wells worked for Genentech, and Genentech was founded on the success of Herceptin. It was like their first big drug. Now, look at the cartoons in Figure 5. So I'll turn to there with you. The Figure 5 in the big paper. And by the way, the last half of the paper is probably not relevant for us, but I just printed it all for you just in case. Okay, Figure 5. You should be able to see what the antibodies are looking like. Look for the little Y structure. And also, by the way, they're using antibody fragments, so you can see a fragment of a Y as well. In fact, if you look on the left side of the figure, you can see the antibodies being used in different ways. This is, once they made their antibody, this is how they say they're trying to use it. And they do some experiments that sort of use it in this direction. So look at those three cases and tell me something about how are they using the antibody to attack cancer. And not necessarily attack, maybe even as a cancer therapy, as part of the cancer therapy. What's a word or phrase that you see that could be a way they want to use this? The BITE-mediated T cell activation, is that what you see? Yeah, exactly. So they can use the antibodies and look, find the antibody there, the color blue, and you see the tumor cell. The antibody is binding to CDCP1 on a tumor cell. They identified CDCP1 with this screen, and so they say we have an antibody against it. What we can do is we can tie another protein to it that will bind T cells. And then we have this antibody that will grab onto this tumor cell, and then it will attract T cells because it will bind to them and say, hey, come over here, check this out, isn't this tumor weird? You know, and then the T cells are activated. That is, that's number E. That is one way they can do it. So yeah, looking up to number B, what do you see there that will be, this one's even more straightforward than E is. What's the antibody doing in B? What's the symbol that you see in part B that you don't normally see? It's conveying.
It's binding it as well as killing the cell.
It's the cause and effect is around, yeah, binding it and as a result killing the cell. Basically you have something toxic, something poisonous, maybe even a radioactive nucleus that's attached to a secondary antibody. And the secondary antibody binds the primary antibody, which binds CDCP1. Now, the one thing to realize about this is they're using 2 antibodies instead of 1. That's because antibodies are special and Fc regions are constant. So actually, we have a lot of antibodies that are like the red antibody. They bind the Fc region of a bunch of other antibodies. Then you can make specific blue antibodies that will do the job you want. They will bind to the cells you want to kill. And then you pull from your library the secondary antibody, which is the red antibody, and that's the one that will bind Fc region. So basically, if the antibody was made with a human Fc region, you can pull out an Fc region binding antibody that is conjugated to the uranium, and you don't have to use as much uranium, because it's hard to get uranium onto an antibody. This is actually one of the more subtle points of the whole paper. The whole idea of using a primary and secondary antibody, because just using a primary antibody and putting the nasty stuff on the blue antibody, that would be expensive and wasteful. This is actually better, because the Fc region is constant, do it on an Fc binding antibody, and then you can use it for other antibodies too. You can use the same secondary antibody with a whole bunch of primary antibodies. That's the idea, and that idea is actually going to come back later in this talk. Okay, and so the final thing that you see on the bottom, this is actually a kind of imaging. Can you tell what kind of imaging is in G and H? They're suggesting that they do it in G, and they're showing the results of it working in H. It's a kind of scan that maybe you've had done to yourself. Do you see what it is? Yeah, it's a PET scan. And I don't actually know what the difference is. I know it's proton emission tomography. I'm not sure the practical difference is between PET and CAT scan, but to us as a patient, it's pretty similar. It's a scan they can do to find a tumor. And so what you do is, apparently zinc 89 shows up in PET scans really brightly. But how do you get the zinc 89 to stick to the cancer cell? Well, you attach it to this antibody. It binds CDCP1, and that binds to the cancer cell. And they have a bunch of the figures to the right show big red blotches, basically where it is going to the tumor. It's highlighting what the tumor is and what it's doing. Okay. So that's what they're wanting to do. That's what they want the antibodies to do. This is the antibody drug conjugate cell killing. Basically, 2 antibodies stick together, they form a little chain, and they bring toxin to the tumor. And the other one is more immunological. We will talk more about T cells on Friday. The main thing here is it brings the T cell close so the T cell can attack the cancer. And then here's the PET scan, and they show that it works. It works in mice at least. That this will go to the tumor and it will make it light up in the PET scan, really obviously. So nice clear diagnosis. Okay, so the thing that you want to look at is you want to look at the end of the paper. So let's flip through now. Oh, actually, we've already talked about this. We were at the very end of the paper. You can see what we already got out of the abstract. So just flip to the end of the paper and find the CDCP1. So flip to the last page and just scan up and down and see how you see CDCP1 over and over again. That was the end result of what they did. They did all this work and they got one target out of it. And they got antibodies that bound that target. So it was a lot of work for one target, and actually this is pretty common in biotech. It's hard. But you can look at this and you can see that they actually ID the target, and they say right here, they go back into literature and they talk about how there are these other papers that says CDCP1 was important to these specific cancers. And these are all RAS related. So they're saying maybe this is a key molecule that is always there when RAS is mutated. One of the problems is you mutate RAS and a whole bunch of other stuff changes. So how do you tell what the important part is that's changing? Once they identified this molecule, they went back and they found other people who had also found this protein in specific cancers. And so they were like, I think we're on the right track. It's also highly expressed. And that's what makes it key. There's a lot of it on the surface of the cell. And in fact, that's what you're trying to find with this. You're trying to find something distinct, but also prevalent. You know, it needs to be a lot of it for us to bind it. So how do they choose CDCP1? How do they make antibodies to it? Flip to this figure. Let's see which one this is. This is where they took this, they screened all the proteins on the surface. Now, it's almost a joke now in biotech, but you call anything an ohm if you're looking at the whole cell part of it. So proteomics, genomics, everyone's familiar with, but it's sort of taken over all sorts of words that way. And what they did is they used, it doesn't say right here, what biochemical technique are they using here? If you look at this figure, what biochemical technique are they using in this figure? Data that looks like this. Light, heavy, M/Z, and then you actually have the abbreviation for it up there. Yeah, so a lot of proteomics, and in fact, here in Seattle we have the Institute for Systems Biology. It's one of the pioneers in using mass spec, because everything has mass. But how do you tell one thing apart from another if they all have mass, right? And you want to be able to tell one experiment from another experiment. The way that you do that is you use mass. You use different isotopes. And so this is probably the expensive and tricky part of this whole thing. You have the cells which don't have any vector, they just have normal RAS. Then you have cells that you put mutated RAS in. So these are cancer cells. And the mutated RAS you feed on heavy nuclei. The empty ones get regular nuclei, or lighter nuclei. I guess I shouldn't say for sure which one's more prevalent. That means that the proteins that come from the light will be a couple protons lighter. And that's enough for mass spec to reliably detect. Okay, so you have the heavy means cancer. If you have more of the kind with the heavier isotopes in it, as detected by mass spec, that means there's more of this in cancer cells, in mutated RAS. Now, the other thing that they did is they also gave the cells drug treatments. So these are both, in this case, both of them have mutant RAS. But then you give one of them a cancer drug, and then one of them you just give nothing. That's the vehicle. And the other, the blue one, you give the cancer drug. Again, the heavy ones are the cases where the cancer drug, and the thing is we have cancer drugs that will turn off RAS, that will ameliorate the RAS problem, okay? So, what they did is they took these, and then they do this thing called the volcano plot. Now, remember what's the protein that we're looking for up here? You want to look for the end result to sort of orient you. Yeah, so can you find that?
Yeah, it's the middle right, and then the one
right there.
left.
Right. So that means that, and you see how it's blue on this side, and it's red on this side. That means that it responded, it was increased in cancer. So this side must be increased in cancer. That was the first side. And this side must be where you just have the drug, but it's increased when you add the cancer drug, and that shows the effect of the drug on things. Basically what you see, and what they're looking for, is not necessarily one way or the other. Oh, well, they are looking for things that will be a blue on this one and red on this one, I think. And you notice how there are equal and opposite things. Basically, if you have more of it when cancer is transformed, and then you add the drug, then you get less of it. That's the idea here. So it's noisy data, it's messy data. You're looking for a needle in a haystack. But you can see that there are several proteins that you find that do equal and opposite effects to a significant degree. These are potential hits. Now notice that these potential hits are still kind of messy, and there's nothing that's like hitting it out of the ballpark, you know? This is not obvious data, but you take a bunch of them and you try to see which of them will do what you want them to. So this is how they did KRAS activation, and then they had 3 different drugs that they tried. Actually, these don't, I might have said these inhibited RAS directly. No, they actually inhibit other problems. They're cancer drugs that attack any part of the cancer pathway. And so they basically want to see where you have something that shows up in the RAS activation. When RAS is activated, you see it on the surface. And when you add the drug, it takes it away from the surface. And there's nothing that responds to all situations. So there's nothing that responds to all of them. There's one of them that fits in 3 situations, and 10 of them that fit in 2. There's 13 basically that do multiple situations at least. That's the shaded area right here. Okay. And in fact, AKT inhibitor, by the way, is protein kinase B. That's something that you also talk about when we talk about RAS and signaling pathways. If you look back at chapter 12, you will see these things over and over again. Okay. Now here's a question I have for you though. You might have thought, a lot of people said, why do we need to go for the proteins? Because going for the proteins is difficult. If you go for the proteins, why can't you just go for the mRNA instead? Because it's easy to take all the mRNA in the cell and to just sequence it. So people did that. And in fact, you have this technique is called SILAC. And the RNA technique is called RNA-seq. And so what they did is they took all the SILAC and they basically said, if it's red, then we see it go down. If it's blue, then we see that protein go up in cancer situations. And let's compare that to RNA-seq. Now I want you to look at the data and look back and forth. If they correlate, then both of them will be the same color. And they sort of arrange it so that you have most of the, they arrange it, they're arranging it by some kind of correlation, but you can see that most of the RNA-seqs are red on the left and blue on the right. What would you say, when you go to the extremes, you see red and blue matching up. But would you say that most of what's going on, where do you see red and blue not matching up? If red and blue don't match up, that means RNA and proteins disagree. RNA and proteins disagree. So it's nice over here. And where do you see it not matching up?
Like, corner
Yeah, so like right here, definitely you got blue and red. And here you got blue and red. Here you got blue and white. Red and blue. Blue and red. There's a little matching here, there's some matching there, but it probably looks like more doesn't match than actually does match, right? So what does this mean? This means Dr. Tinlin likes to do things with RNA, but you can't assume that just because you have a lot of RNA in the cell, that RNA proportionately produces more protein on the surface. This is true that there's more RNA in the cell for these things, but for this thing, there's a lot of RNA in the cell for these blue lines, but then there's not much of it on the surface. So there's not a direct correlation. And in fact, for a few things at the extremes, there is correlation. And this is the kind of thing where experiments can be valid. But there's a big range in the middle where the experiments are not valid. The correlation is not valid. If you want to know what protein's on the surface, you can't just look at RNA. That's what this means. I think do I circle something or something like that? I don't know what I did. Yeah, whatever. Okay. So now pull out the other one and look at this, because I want you to have this as well. And part of the point of this whole lecture is to orient you to these so that then you can like take them, you can investigate them and use them. Open this up. Oh, actually, I'm sorry. We're not at this one yet. We're still on the big paper. And let's find this particular thing in the big paper. There it is. It's figure 2, page 6. This should look a little familiar if you remember Delano and Wells. And you don't have to have the Delano and Wells paper for this next quiz, but you should have this along with you. But this is the idea that they did. And this is where they talk about, over here, this is all biotech stuff about how the stuff actually works. But I want you to focus on this right here because this is phage display. Everything we talked about with Delano and Wells applies to this. Instead of doing a 20-mer peptide, what they're doing is they are mutating selected residues on a fragment, an FV fragment of the antibody. But they are keeping track, they're binding it to something, they're keeping track of what the sequence is by sequencing the RNA inside the phage, and then they're doing multiple rounds. All of that is exactly like what happened in Delano and Wells. So I want you to see the connections between those. By the way, and he said this when he presented it in person, this is where I got the idea. He said something that isn't in the paper. They had to pick which FAB fragment they would use. And because of his extensive experience with antibodies, there was just an old fragment that was just a scaffold that expressed super well, like the bacteria just loved to make it for some reason. And that's the kind of thing that's not in the paper, but it's crucial to the success of this. You have to have a scaffold that the bacteria like to make. Now the other thing about the scaffold, and by the way, the scaffold is put on an FC domain, but if you remember from the antibody lecture, FAB fragments are different from FC fragments. These are actually different antibody classes, and they just splice them together with a proteolysis site in between. And they use that for the whole thing. That's part of what the robot is doing. So this is where the antibot comes in, and this is where you look at the second paper that you have right here. Okay, so just check out this paper and realize that there's a connection here. Flip this paper to the most important page, which is the second page. And I want you to look at that. So on the top of it, they have this figure, which is exactly like the phage display that we talked about. Again, this is the concept. Probably if we have a concept coming up over and over again, I might ask you to explain it on a quiz. But the other thing is my question, this doesn't even get into the paper. But if you look at it, where exactly are they mutating and how are they mutating it? And realize that this is the thing, this is where you can see the stuff that looks like Delano and Wells. There's the whole antibody fragment. They're only mutating loops on the top of it because those are the only parts you have to mutate. But even then, remember our problem from Delano and Wells. There's no way you can cover all of the possible sequences, so you have to choose your battles. And what you end up doing is you end up mutating only these residues, so you see that there's a handful of residues, and then they even limit them a little bit. You see that, so YSG, they tilt it toward being Y, S, and G, and just to remind you of your amino acids, tyrosine, serine, and glycine. By the way, do you remember when we were looking at the sequences? Do you remember which amino acid is my favorite from the last lecture that I did a paper on about how cool it is at binding? And we also saw it in antibody CDRs. It's one of the ones up there. And it's not serine or glycine. So, tyrosine. So because antibodies love tyrosine, yeah, basically they're doing a bunch of tyrosines. And then they have other options. You know, there's 50% a mixture of any amino acid. They tilt the scales. And so there's a 50% chance that these will be, or more, that these positions will be Y, S, or G. And then they mix in about 50% chance that it could be anything else. And that's how they narrow down the scale so that it actually works. Tyrosine is the best. Maybe I should just ask that on the quiz. Which amino acid is the best? If you don't remember, it's a 1 out of 20 chance. But hopefully you'll remember. If there's one thing I want to get across in this class, tyrosine is cool. They actually calculate how much diversity you could get. And so even with this, the total amount of diversity, if you wanted to produce one of every kind of antibody, you would need 10 to the 30 different antibodies. They actually can produce 10 to the 10th. So even this is orders of magnitude below the truly exhaustive sequence search. But the question is not, is it exhaustive? The question is, does it work? Do we have enough diversity to generate an antibody to this unknown surface? And because they found it, they do. Okay. So they mentioned ELISA, and I think I talk about this in biochem, but this is another thing that you may or may not have gotten from another class. How comfortable do you feel if I ask you to explain what ELISA is? Have you encountered it in other classes at all? Okay, so not much? Maybe a little?
I have, but I don't remember much of it.
Sure, okay. Well, that's why learning is a spiral, okay? So we're going to go back and forth. But if you look at this on this certain page, I don't have which page this is on, but if you look at the methods for this, you'll see that they're using ELISA. And if you look at that antibody thing again, you have 96 well plates, or maybe 24 well plates, but you have these plastic plates. Did we ever use those in class? The crystallization trays are close, but they're for crystallization. Have you ever used 96 well trays? I probably should have taken one to show you, but it's like a protein crystallization tray. It's just got 96 wells in it, and you can do 96 different experiments by pipetting into it. That's what the robot was doing. The reason why the trays, plastic trays, work well for this is because you're using the bottom of the tray as part of the experiment. So instead of binding to a column, you're binding it to the bottom of the tray. And that's what ELISA is. So let me explain a little. This is where we screened them by phage ELISA. They could tell what was bound and what was unbound. They collected what would bind by ELISA, and then they sequenced the DNA of what bound, and then they could enrich it, and they could do it again, exactly like Delano and Wells. So this is what ELISA is, and this is one of those things that you want to be able to take out of your education to bring into the real world. ELISA is where you have an antigen stuck to the surface of the plate, and then you bind antibodies to it. And this is the same thing, primary, secondary antibody, the same pattern that we had before. You have a primary antibody that binds the antigen, and it has a very different variable region, but the FC region is actually constant according to the species and type of immune cell that made it. That means that you can pull out of your cabinet a secondary antibody that binds the FC region, but this one is coupled to some kind of payload, and they literally call it a payload sometimes. In this case, it's not something toxic, it's not like uranium or something like that. What it is it's just an enzyme that changes color. The enzyme catalyzes the conversion of an uncolored substrate to a colored substrate, or it produces color somehow. When you see the color, you go down the chain, you know you have the antigen. Red color means blue antigen is in the plate. And more red color means more blue antigen is there. So ELISA is really important and cool for this whole process. And so you actually they have products that they're trying to sell you ELISA and things like that. And this is where you can see what it actually looks like. 96 well plate or a 384 well plate if you really wanted to get fancy. And the antibody, something binds to the well, you add your sample, you wash it, you add your secondary antibody, you wash that, then you add your substrate and it turns colors. This is what the results look like. You can see this is a 96 well plate, sort of zoomed in. And where you see the yellow color is you must have your antigen in those wells. You can also use it for blotting, which is a kind of thing that you do. You blot an SDS phage gel that you've run. So in this case, you have an antibody that's binding to something on the bottom of the well there. In this case, you have an antibody that binds to one specific band on this gel. And you can see that here's a purification, and you're getting more and more pure as you go across. But really, when you have this many bands, how do you know that the protein that you're after is actually in that mess? Well, if you have an antibody against it, the antibody can bind to it and then turn the immunoblot dark at that point. So you see that in the lysate, there is the protein that you're interested in, but not as much as when you purify it more. You get rid of other proteins, but you're actually keeping the one protein you're interested in. This is a nice proof that you're actually doing what you want to do in the lab. And so there's sort of jokes about this. So see what a Western blot looks like. And then they're trying to sell you Western blot reagents, results with reagents from Thermo Fisher, a nice single band, and that's what was in the textbook, right? When you're working within the lab, you know protein markers look like this, and you know that the textbook example is like this. But a lot of times you'll have bad antibody, which is going to give you extra bands. If you have a specific antibody, it's only going to stick to one thing. But there are some antibodies, and they want to sell you specific antibodies. That's part of what they're selling you. But sometimes you have a bad antibody that sticks accidentally to other stuff. And then, of course, they make jokes about this. Protein markers, and then you have felt tip markers who make this. If you have Western blot bands over here, then you have band practice here and frowny bands there. They're always trying to make you jokes, but of course you know that they're trying to sell you something too. But, maybe this will help you learn what a bad situation looks like so you can compare it to the good situation where you have a strong single band. And so you can use this on any scale. If you can use it with 384 well plates, you could use it with a 4,500 well plate in a sense. This is basically a 4,500 word well plate where they have all the proteins from the malaria parasite and they put them on the plate. And they make each of them, they basically run ELISA so that the ones that you have antibodies to will light up. This is from I've been to Africa, but I don't think I've been infected by malaria. So probably my blood, and definitely I haven't been infected lately. So definitely if you run my blood over this protein array, you get a lot of dark spots, a lot of nothing lighting up. Because I don't make antibodies to malaria just for the fun of it. Your body doesn't waste energy like that. But if you take somebody living in West Africa who is infected by malaria and has probably been exposed to malaria all their life, you can see that many of these 4,500 proteins, there are antibodies that they have to like thousands of these proteins because you see them all lighting up all over the place. And this is all, by the way, this is why a secondary antibody is helpful, because you don't want to label every single antibody. You can't label every single antibody in that person's serum. But you can bind it to the plate and then have another antibody that binds all of them, and that antibody can be labeled. It's better to label one antibody than to label 1,000, and it might not work anyways. So that's why I want you to know how the secondary antibody thing works. By the way, if we ever do a CRISPR Journal Club, we sort of turned that, I remember it was one of the options that we had on the beginning of class. And if you ever need a paper for a CRISPR Journal Club, you could actually pull this paper out because they use CRISPR, at least for part of it, because they talk about, they do a CRISPR screen to back up their mass spec screen that they did, the SciLac screen. We aren't going to focus on that because we're not focused on CRISPR, but I just want to say it's here, and maybe someday there will be a paper that we talk about in class that will be useful for another class. But basically the CRISPR screen and the mass spec screen both ID just two proteins, and here they are right here. This is near the end of the paper. ITGP5 and CDCP1. In fact, let's find it. So let's flip to the end of the paper, pull out the Elife paper, and it's not really the end because they have so many methods. You might want to actually, in the methods, you might want to actually see where they have like phage display and like circle that so you can find it more easily. But more than that, I just want to go, it should be near the end but not at the end. Oh, actually, it might, I might be lying. It might be at the end for the discussion. Yeah, but it's actually in the middle because it's the middle of the thing. So it's good to look through the paper and to try to find this. Let's try to find this. It's after the CRISPR screen section. So I found the CRISPR screen section on page 10. I'm turning pages. I'm looking for. It'd be funny if it was actually at the end after all that. Oh, perfect. Okay, that's good. Yes, there it is. So look for the paragraph starting remarkably, and go down to where it says ITGB5 and CDCP1, and circle that. That is the result where both screens go together. Remember there were 13 results that showed up from the first screen? And so they did the CRISPR screen, and only 2 of them were in common to the 2 different types of screens. But that's something. And so the question is, and then they ran with the CDCP1 because CDCP1 has been found by other groups to be important, and that's where they got the end result from. Now the one thing I don't have here, my question is, why didn't they move on with ITGB5? And I'm scanning and I don't see ITGB5 anywhere here. Oh, okay. Yeah. I think they just decided to go for the one and not the other. They said they know that the CDCP1 is often associated with this particular integrant. And integrants are sort of like the concrete girders of the cell. Okay, they're or the cell surface, I should say. They're very common. And so the fact that they are showing up with these, it's suspicious. It's not very specific. Because integrants are everywhere and they don't want to stake their whole lab on that. CDCP1, on the other hand, is a unique molecule. It's distinct. It's not this kind of generic molecule. And so therefore they want that. They don't really tell you that. They just sort of say, they have some line about how CDCP1 is integrin associated with this particular integrin. And so they don't actually say that we're going to go the other way. So that's what you want to do. This is actually what I have for you on this particular paper. I guess any questions about the paper or things like that? I sort of would want you to have the paper in front of you, and I realize it would take a long time to leaf through it. And so what you want to do is you just want to review the paper sometime while this is fresh in your mind and make some kind of notes to transfer this. I will record this so that you can actually return to part of it if you want to. But yeah, make your paper the paper copy a resource for you to use.